In this video, we're going to learn how fungi reproduce. Before we get too far into talking about fungal reproduction, let's do a quick review of the concepts of haploid and diploid. You've probably heard these terms before when you learned about mitosis and meiosis. Haploid means an organism has one set of chromosomes. Whereas diploid, di means two, diploid means that an organism has two sets of chromosomes. We call these paired chromosomes. And each chromosome in the pair is a homologous chromosome to the other one in the pair. So the pairs are homologous chromosomes. These homologous paired chromosomes come from sexual reproduction. So during fertilization, a male gamete fuses with a female gamete, and it forms a diploid zygote. Now, each of the gametes is haploid, but the zygote is diploid. And we kind of denote diploid as 2N, whereas haploid is written as 1N. So the male gamete is 1N or haploid, the female gamete is 1N or haploid, and then when they fuse together, it forms a diploid zygote. So each set of the two sets of chromosomes comes from a different parent. Now, in organisms like fungi, we don't really have a mom and a dad or a male and a female, but we do have a set of DNA or a set of chromosomes that comes from one organism, and then a genetically different set of DNA chromosomes that comes from a second organism, and they fuse together to form this diploid zygote cell that then develops into a diploid form of the organism. Both haploid and diploid organisms can go through mitosis. But only diploid organisms can go through meiosis. Another thing to keep in mind is that during mitosis, the ploidy of the cells produced is going to be the same as the original cell. So for example, if a haploid organism goes through mitosis, so they start out as 1N, the daughter cells are also going to be haploid or 1N. If a diploid cell goes through mitosis, it starts out as 2N and it ends up as 2N. With meiosis, the ploidy is going to decrease. So in meiosis, the cell is going to start out as 2N, but it is ultimately going to end up as haploid or 1N. And this is because those paired homologous chromosomes get separated during meiosis, so that all of the daughter cells produced now only have one set of chromosomes as opposed to the two pairs or sets that they started with. Fungal reproduction may be asexual or sexual. Asexual, A means without. So this is without sex or without fertilization. And there are three different types of asexual reproduction in fungi that we're going to talk about on the next couple of slides. And those include fragmentation, budding, and spore production. And these all rely on mitosis as a way for the cell to then create more cells, or for an organism to create more organisms like itself. Now, sexual reproduction actually includes this kind of alternation of generations in which sometimes the organism will reproduce asexually and other times it will reproduce sexually. And the main difference here is that at times the organism is going to go through mitosis, and then at other times it's going to go through meiosis. And so the ploidy is going to change. So at times the cells in the organism are going to be haploid, and at other times the cells in the organism are going to be diploid. Now, this is different from what happens in humans. So in humans, our kind of primary life stage is going to be diploid. So the majority of the cells in a human's body are diploid. The only time that there are haploid cells are when they produce gametes. So when cells go through meiosis to form either eggs or sperm, depending on whether you're male or female. These are the only haploid cells in the body. And these gametes are not very long-lived and they're not independent organisms. So these are only cells that are involved in reproduction. During fertilization, during sexual reproduction, they fuse together to form a diploid zygote, which ultimately forms our main kind of life form of this diploid organism. Now, if we go back to fungi, there are going to be times when the organism itself, all the cells are going to be haploid, and then at times when all the cells in the organism are going to be diploid. So they actually have a kind of multicellular form that is haploid, whereas in humans we don't have that multicellular form, we only form these haploid gametes. So the first form of asexual reproduction we're going to talk about is known as fragmentation. And so what happens, if you recall, we talked about having this kind of mushroom, and then we have the hyphae, which are these kind of string-like things underground that form the mycelium. Remember that these hyphae are made up of cells. So this is a hypha, and altogether this is a mycelium. Now during fragmentation, what happens is that a section of a hypha can actually break off, and then those cells can go through mitosis in order to make more cells, which ultimately form a new mycelium, which ultimately results in a new fungus altogether. Budding is a form of asexual reproduction that happens in yeast. And remember that yeast are the unicellular fungi. So you can see in this picture here, this is a picture taken through a microscope. And you can see an example of budding happening right here. So first we have the parent cell here. And the nucleus of the parent cell has gone through mitosis. So the DNA has been copied and then the chromosomes have been separated to form two new nuclei. During cytokinesis, this bulge is forming here, and this bulge is going to be the bud or the offspring. And eventually that bud, that daughter cell, is going to detach from the parent and become its own yeast cell. And that might be what we're seeing right here. So this could be a bud that has already detached recently from the parent cell. Another way that fungi can reproduce asexually is through the production of spores. And these spores are going to be asexual spores. And I say that because there are also sexually produced spores. So right now we're talking about the asexual spores. And what happens is that in the mycelium, and remember the mycelium is just this network of hyphae that all together make up the thallus. And these are going to be those cells that are primarily underground with the fungus. And so these cells go through mitosis. Remember that mitosis is a form of asexual reproduction. And they form these spores. Now it's important to note that these spores are genetically identical. And they are genetically identical to each other and genetically identical to the original cell that they were produced from. And this is important because genetic diversity is typically considered a good thing in unstable environments, but having less genetic diversity can potentially be beneficial in more stable environments. And we'll talk more about why that is later on in the semester. So the spores produced are then going to be released into the environment, and they might be released a number of different ways. They might be carried on the wind or on an animal or through water. You can see this picture here where the spores are being kind of released out of this puffball into the wind for them to be carried to another location. So germination occurs when that spore lands in an area that is hopefully beneficial to its survival. So if that spore lands in an area where it has the nutrients and environment that it needs, it can start going through mitosis in order to form a new mycelium. And then that new mycelium can ultimately produce a new fungal organism. And I should mention that sometimes these spores are released within a sporangium, and a sporangium is just a protective sac. And this sac might help to protect the spore from things like dehydration, heat or cold, or potentially digestive enzymes if it's passing through the gut of an animal or something like that. Some fungi reproduce sexually, and one benefit of sexual reproduction is that it helps to increase genetic diversity, and this can be especially beneficial in adverse conditions. And this is because the more kind of difference you have in genetics, the more likely you're going to have some individuals in a population that are able to survive whatever is happening in the environment. So genetic diversity isn't necessarily, it's not beneficial for a single individual, a single mushroom, a single fungus, but it would be beneficial to the population of fungi in the environment so that if a big change in the environment happens, at least some of the organisms will survive in order to be able to reproduce and repopulate the area. So we mentioned that some fungi can actually switch between asexual reproduction and sexual reproduction. So asexual reproduction is typically faster and easier in the sense that it takes fewer resources in order for an organism to reproduce asexually. However, if the environment is changing, then it might be worth the organism to invest additional resources to reproduce sexually in order to increase that genetic diversity. So in fungal sexual reproduction, there are two genetically different mating types involved. And in fungi, we call these types positive and negative. This is kind of similar to what we see in humans, where we have male and female. But in humans, males are genetically XY, meaning that they have an X chromosome and a Y chromosome, and females are XX, meaning that they have two X chromosomes. There are also structural differences between male and female with regards to internal and external genitalia. With fungi, we don't have those differences. We don't have XX and XY chromosomes, and we don't have these kind of genitalia type structures that would make one be considered male versus female. Some sexually reproducing fungi are homothallic. Homo means same, and thallic refers to the thallus. Remember, the thallus is made up of the mycelia, which are made up of the hyphae. And so what homothallic means is that both mating types, so cells of both mating types, are present in the same mycelium or thallus. And so essentially there are, when we say both mating types, remember that it means that we have positive mating type cells in the mycelium, and we have negative mating type cells in that same mycelium. And these fungi are self-fertile, so they are able to basically do sexual reproduction with themselves. Heterothallic, hetero means other or different. And thallic, once again, has to do with the thallus. And so in a fungus that is heterothallic, two different mycelia have to be involved in order for sexual reproduction to occur. So you would essentially have your kind of your mushroom here with its thallus made up of these mycelia. And then you would have to have another mushroom with its mycelia making up the thallus. And one of them would have to have the positive mating type cells in their thallus, and the other one would have to have the negative, and those cells would have to fuse in order for sexual reproduction to occur. If we go back to homothallus, so homothallus would be where you essentially have one mushroom with its hyphae making up the thallus, and some of the cells would have the positive mating types and some would have the negative, and they would be able to merge or combine in order to do sexual reproduction. In this diagram, we're looking at kind of the generic life cycle of fungi when a fungus switches between asexual and sexual reproduction. Now, when we talk about the specific phyla of fungi, there might be some variations to this life cycle that we will talk about, but this is a good kind of generic general start. We actually talked about the top part of this diagram earlier in our video. This was when we talked about the asexual phase of the alternation of generations. So to do a quick recap, remember the mycelium, which is haploid, is going to go through mitosis in order to form haploid spores. And these haploid spores are genetically identical to each other and to the original cells that went through mitosis in the mycelium. If a spore lands in an area that is conducive to growth, it will germinate, which means that it's going to go through mitosis in order to form this multicellular mycelium, which is ultimately going to develop into a new fungal organism. So the bottom half of this diagram is looking at the sexual phase of the alternation of generations. And remember that some fungi can switch back and forth. Depending on the environmental conditions. Now, different phyla are going to have some different specifics when it comes to the sexual reproduction stage of this alternation of generations. However, they all have the same kind of 4 basic steps to them. So the first step is plasmogamy. followed by karyogamy, followed by meiosis, followed by germination. And we're going to talk about each one of these steps on the following slides. The first stage of fungal sexual reproduction is known as plasmogamy. And this word translates to cytoplasm marriage. So you can think of plasmo referring to the plasma membrane, which is also the cell membrane. And so essentially the cell membranes of two fungal cells are going to fuse together, and then the cytoplasm and all the contents inside those cells are now going to be encased in one single cell membrane. Now each of the cells involved in this plasmogamy are going to be haploid. Remember, haploid means that they are 1N, meaning that they each have one set of chromosomes. So if we were to kind of draw this out like a cartoon, we would have cell number 1 that's going to have a haploid nucleus in it. Cell number 2 that's going to have a different haploid nucleus in it. And what's going to happen is they are going to fuse together to form kind of one big cell that now contains two separate haploid nuclei. So this is actually different than what happens in fertilization. In fertilization, one cell fuses with another cell and the genetic information fuses together right away. In plasmogamy, the cytoplasm is fusing together, but the nuclei are staying separate from each other. This ultimately forms a cell called a dikaryon. Di means two, and so this tells you that there are two nuclei. And we would denote this as N plus N because you've got one N nucleus here in red, one in blue, so now you have an N plus N ploidy. This is not the same thing as 2N because 2N would indicate diploid, which would be the sets of chromosomes being in the same nucleus. In this case, we still have haploid nuclei, and each set of chromosomes is still being housed in its own nucleus. And you can see this on the diagram here with this picture being drawn right here. This is the same picture that I recreated out here. It's also important to note that a dikaryon can also be known as a heterokaryotic cell. This heterokaryotic dikaryon condition can actually exist for a really long time, including days, weeks, months, maybe even years. And this allows the dikaryon to essentially wait until the environmental conditions become beneficial for it to then move into the next stage of sexual reproduction. The second stage of fungal sexual reproduction is known as karyogamy. And if we look at this word, karyo should make you think of nucleus and chromosomes. And gamy is marriage, as we learned on the last slide. So karyogamy can be translated to nuclear marriage. So remember that during plasmogamy, there were two cells, each with a haploid nucleus, and the cell membranes fused together, which united the cytoplasm, but we ended up with this dikaryon cell that still had two separate nuclei in it, but it had these two haploid nuclei. So remember, throughout all this, these nuclei are haploid. Now in karyogamy, what happens is that within that cell that was that dikaryon that was produced during plasmagamy, those two haploid nuclei are now going to fuse to form a diploid nucleus. Now remember that at the end of plasmagamy, we had a dikaryon. And now at the end of karyogamy, we have a zygote. And we can see that on this picture over here. This is our zygote with the diploid nucleus in it, whereas the dikaryon had two haploid nuclei. And remember that fungi are eukaryotes, so these nuclei are membrane bound. So there's a membrane surrounding the chromosomes in each of the nuclei. And so it's actually the nuclear membranes that are fusing, bringing together the genetic material from the two separate nuclei and forming it into one nucleus that contains two sets of chromosomes. So this process has led to a genetically different or unique set of chromosomes that are now together in the zygote. Now, if we were to draw this out, remember that the chromosomes in eukaryotes are bar-shaped. So we could say that the chromosomes in the red haploid nucleus over here are going to be bar-shaped, and I'm just going to draw 3 of them for now. The one in this blue cell here would be the same number of chromosomes, and really these chromosomes are very similar to each other. They have the same types of genes on them, but they might have slightly different variations of those genes. Now in the zygote, these chromosomes have come together so that there's now these paired chromosomes all contained within the same nucleus. In the third stage of fungal sexual reproduction, we have meiosis. And I'm not going to go over all of the different steps of meiosis. You should have learned this in a previous biology course, but we are going to cover kind of the overall basics. So remember that we're starting with a zygote. And the zygote is diploid. So it's going to have a set of chromosomes that it got from one original parent cell and a set of chromosomes from the other parent cell that went through plasmagamy and then karyogamy to produce this zygote. So I'm just drawing 3 blue chromosomes and 3 red chromosomes just as an example. Now this cell is going to go through DNA replication and then a couple of rounds of cell division. And ultimately, it's going to form 4 daughter cells. And each of these daughter cells is going to have a nucleus. But each of these nuclei are going to be haploid, or 1N. So we are changing the ploidy from 2N to 1N. Now during this process of meiosis, essentially these chromosomes are going to get kind of shuffled and then dealt out into these daughter cells so that the daughter cells are going to be genetically different from each other. So to represent that, we could say that this one daughter cell maybe ends up with 2 blue chromosomes and a red one. Maybe this one has 2 red chromosomes and a blue one. This third one maybe ends up with 3 blue chromosomes. And the fourth one maybe ends up with 3 red chromosomes. These daughter cells produced by meiosis, so once again these arrows here are representing that process of meiosis, these are producing the sexual spores. And we can see those on the diagram over here. Now, this is different from the spores that we made during asexual reproduction. The asexual spores are genetically identical to each other and to the parent cell that they were formed from. But these sexual spores are genetically different from each other and genetically different from the original cells that they were produced from. And remember that these spores are haploid, so 1N, and they were produced from a zygote that is diploid, which is 2N. Now these spores may develop in a structure known as a gametangia, which is essentially acting like a gonad in fungi. So if you think about in humans, females have ovaries that produce eggs, and males have testes that produce sperm. Well, in this case, fungi have gametangia that produce these sexual spores. The fourth step of fungal sexual reproduction is germination. And germination occurs when these spores are released into the environment and they hopefully land in a suitable environment for them to survive and grow. Now, spores can be disseminated in different ways. They can be carried on the wind, they can travel through water, or they might hitch a ride on or inside an animal. If the spore lands in a suitable habitat, that spore is going to go through mitosis in order to make more cells that are ultimately going to result in the formation of a mycelium. And then this mycelium is going to keep going through mitosis in order to form a new fungal organism. So as we mentioned before, some fungi can switch between asexual and sexual reproduction. Now, the reasons why they might favor one over the other has to do with what's going on in the environment and also access to resources. When the environment is stable, then the fungus is actually going to prioritize asexual reproduction. Now, the reason why this is that there are fewer resources and energy needed in order to reproduce asexually. Organisms also don't need to find another organism with the opposite mating type in order to reproduce asexually. We also know that with asexual reproduction, there is not much genetic diversity happening, or the genetic diversity is not increasing. And that's okay in a stable environment because we know that the organisms that are surviving and reproducing have the proper genetics to survive and reproduce in that environment. However, if environmental conditions become unstable, a fungus might switch over to sexual reproduction because it helps to increase genetic diversity. Because remember that during plasmagamy and karyogamy and meiosis, the organisms are essentially bringing together different genetics, shuffling them, and then kind of dealing out these genes into the spores. And this helps to increase or kind of change the combination of genetics that are found in the new organisms that are being made. Now, genetic diversity does not help a single individual, but it helps a population. And what this means is that If a population has a lot of genetic diversity, if something detrimental happens in that environment, there are likely to be some organisms that have the right combination of genetics for them to survive and reproduce and maintain the population. Other organisms in that population might not have the, quote unquote, right genetics, and they may die off. So it's not really helping the individual, but the population or the group of fungi as a whole will survive whatever is happening in the environment. Now, you may wonder why organisms wouldn't just always do sexual reproduction, but the problem is it takes more energy and resources in order to reproduce sexually. An organism also has to be in the same area as a mate with the opposite breeding type in order to reproduce sexually. So if there's no mate around, then you don't have the option of reproducing sexually.
All right, so let's fill in a blank diagram of fungal reproduction just to help us kind of review what we've talked about on the last several slides. So first of all, this picture in the middle here, we're looking at a mycelium. Remember, the mycelium is haploid, so we can designate that as 1N. And remember, the mycelium are just made up of each of these individual strands called hyphae. And the mycelium is also sometimes referred to as the thallus. Now, when a fungus is going through asexual reproduction, this arrow is going to represent mitosis. Remember that mitosis is a form of asexual cell division. And so what happens is that these cells in the mycelium go through mitosis and they produce these asexual spores. These asexual spores are haploid, so 1N, they have one set of chromosomes. And remember, they are genetically identical. And they are genetically identical to each other and genetically identical to the original mycelium. Now, these spores can then be released into the environment and they're going to land on different surfaces. And if they find a surface conducive to growth, they might go through the process of germination. And during germination, essentially what happens is that those spores are going to, in this kind of favorable environment, they are going to go through mitosis in order to form a new mycelium. And remember that there are benefits to asexual reproduction. So the fungi don't need to find a mate. All right, so the fungus can reproduce on its own. It doesn't need to find another fungus in order to reproduce. There are fewer resources needed compared to sexual reproduction, so less energy and resources needed. Genetic diversity also isn't needed because if this is a stable, favorable environment, then we know that the fungi alive in this environment are well suited to that environment. So the genetic diversity is not needed. The genetics that are already there are working just fine. And it might actually be genetically, or it might actually be beneficial to the population to not have genetic diversity because the genetics currently in the population might be the best genetics for that environment at that time. Now, when the fungus goes through sexual reproduction, we have some more steps that occur. So the first step is going to be the plasmagamy. And this is where you have cells from a positive and negative mating types fuse their cell membrane and cytoplasm. And what you end up forming is a dikaryon, which we can also represent as N plus N. And this is also referred to as a heterokaryotic cell. So you can see that we have two nuclei in the cell, and these are two haploid nuclei. In stage 2, we have karyogamy, in which the nuclei fuse. So you have the two haploid nuclei fuse in order to form a diploid nucleus. And this forms a cell called a zygote. And we represent the zygote as 2N because now we have two sets of chromosomes in this one nucleus instead of having two nuclei with a haploid number of chromosomes. The third step is meiosis. And in meiosis, the diploid nucleus goes through a couple of rounds of cell division and ultimately produces daughter cells with haploid nuclei. And these daughter cells are the spores. So the spores here are 1N, so they are haploid. And these spores might be produced inside a specialized structure called a gametangia. Once the spores are produced and released, germination may occur. And germination just means that the spores are going to land somewhere in the environment, and if the environmental conditions are conducive, the spores are going to go through mitosis to produce a new mycelium. So the benefits of sexual reproduction is that we do have an increase in genetic diversity. So this is really helpful when the environment is changing. But sexual reproduction has the downfalls of a mate or an opposite mating type needs to be involved, and it requires more energy and resources. So there are the trade-offs between increased genetic diversity, which may help the population survive in a changing environment, but in order to do sexual reproduction, you have to have more energy, more resources, and more individuals in order to sexually reproduce.